Hello, Biosci 102. This is Dr. Giorgi with lecture 11 for spring 2021. We're going to talk about specimen prep. So you might have seen in your syllabus that it says IHC. That's a type of specimen prep. I thought I'd start with the broader view, which is a bit of a reminder at this point, hopefully. So when you're using transmitted light techniques, your specimen has to be thin, right? And some things are naturally thin. For instance, a layer of cells, they're about maybe 10 microns tall, somewhere in that ballpark. If your specimen isn't already thin to start with, then you're going to cut it, basically. So you use a microtome on a specimen embedded in paraffin, wax. And you're a histotech. You can use a cryomicrotome or cryotome actually for a specimen that's put in OCT instead of in wax. And then throughout your use of a microscope, for instance, when you had them at home, you may have come up with some creative ways to do things. And there's also an ultramicrotome if you're doing things for EM where you're having to cut them nanometers thin. It's kind of crazy, but it's done. So creative ways, well, you can put something in under a dissecting microscope. That's really what it's for, is to be able to work and dissect your specimen and maybe use some sharp razors, forceps, whatever. Sometimes some people creatively freeze things and then smash them into bits, or you can do a squash mount where you literally squash the thing. You're losing a lot of the structure at this point, right? So it just depends on what you're trying to see. You can pull out depression slides, which are some of my favorites. You can just put a fatter specimen in there, but then you still got to deal with the light being able to penetrate. And then whatever you may have come up with, there's just a lot of ways to prepare a specimen. So when we say thin, the specimen needs to be thin. How thin are we talking? Well, if it's reflected light microscopy, it can be a little bit fatter in the sense that we can look at specimens that are millimeters fat. We can look at entire insects. Quite honestly, we can look at anything using a dissecting microscope. I can put my finger in there. As long as you've got the working distance on it, it's fine. So you can see a lot of things. That's the point. It's really easy. You just put your specimen right there. And dissecting microscopes tend to be low mag. I've shown you on this slide the Leica dissecting microscope. I just noticed it has a little Wi-Fi signal because we actually have two of these. They are Wi-Fi enabled, so they send the image to whatever Wi-Fi device you have nearby, but you still have oculars, so it's good. We still like this scope. These are very, very fun scopes. I've drawn in little red arrows to show you the light is coming from its FB illumination. It's coming from not quite the top of the specimen, but from the side. And that there's a specimen in green there, could be anything. And then the light bounces off of the specimen and comes up to the ocular or the camera. And that's what a dissecting microscope does. That's a reflected light pathway in a dissecting microscope. Most dissecting microscopes also have a transmitted light pathway. In other words, there's light that comes from below your specimen, goes through, and you can toggle between the light sources and play around even with illumination angles and just see whatever works best on your specimen. Then there's fluorescence microscopy, right? It's a reflected light technique in that the light is on the same side as the side that you captured it on, so on the camera. However, when you're doing fluorescence, you still need a really thin specimen. You can see only the surface of what you're looking at, basically, because the light is not going to penetrate that deep. And it's also not going to come out from your specimen. If your specimen is thick, the light's going to be trapped in there. And we're talking specimens that are microns thin, basically. Now, of course, if you have a confocal, you can have something a little fatter. That's kind of the point. You can, as long as you can get the light into the center of your specimen, or maybe you don't care, you just, go down a few hundred nanometers from the edges. It really depends on the wavelength and the power of your laser, or, how much light you have. And then whether the emitted light can escape the specimen without being absorbed. And of course, there's all sorts of specimens that are developed for microscopy, for studies in microscopy and genetics, and partly because they have transparent embryos. So something called zebrafish, is a actual fish that's known as zebrafish, has a transparent embryo. So it's great to study development of cells in it, because you can fluorescently label some of the cells and then just make movies, and the rest of the embryo is transparent. There's ways to make embryos transparent. There's a technique called clarity, which removes all the fats and then makes something that's not transparent. So a few micron to a few hundred microns, it just depends on your specimen, right? And whether, how much of it is transparent. There is another technique called expansion microscopy, where they actually blow up the specimen, but that's fixed. It's not for live stuff. And you blow up the specimen, but it keeps the same architecture, the same structure. And they do it using something that is in diapers, actually, the absorbent stuff in diapers. Literally, they took their kids' clean diapers and played around with them. Okay, so, and then a 2-photon or multi-photon scope says that it can go 300 microns or even a little bit more deep, because it's using really long IR light, so infrared light, 900 nanometer light. And the longer the light, the less high energy, the more it can penetrate down into a specimen, and also, not kill your specimen. So multi-photon is about, somewhat thicker specimens. It's a whole technology. It's a whole new $600,000 scope. And the entire purpose is to go maybe an extra 100 nanometers down into your specimen, and also to be able to be more gentle and not fry it as you're looking at it. Excuse me. So in bright field microscopy, our specimens can be a few microns, 4 to 5 microns is what the histotech is usually cutting. So this is for transmitted light. Now the light needs to go through the actual specimen. Again, it needs to be thin. And other techniques, DSE, phase, dark field, whatever, same general ballpark of a few microns. I'm being a biologist here. Do I mean 10 or 300? I mean more on the 5 to 10 to 20 microns, something like that. Okay, so you've got your specimen thin enough. Now, can you see it? The other, okay, it's thin enough for the light to go through, or it works on my scope or whatever. But does it have contrast? And this is where you'll add a stain to it, such as H&E, amatoxylin, and eosin, which are actually, I say H&E because that's what's said, but as you'll learn in your histotech classes, there's a whole wide variety of H's and E's, so that's actually more of a category of stains than an actual just stain. So in general, histotech uses those, and you're going to use transmitted light scopes. And then here we come to IHC /IIF/IF. I'm going to explain these acronyms in a moment, so just hang on to them for now. And the power of this specimen preparation is that it is specific for certain molecules, usually proteins, but it could be any molecule in your specimen. And we're going to deploy antibodies to the rescue, as that's a role in the body anyway. Now they're going to rescue our images. They're going to give us the power to see things, and also enzymes sometimes. And you use these on transmitted light and or fluorescent scopes, depending on what you're doing. So, and just a reminder, of course, you can get contrast without stains if you have a contrast generating technique on your scope, such as phase or DIC. Then you can look at unstained specimens, both live and fixed. Anything where you're staining it, probably fixed. Coming back to this slide for a second, H&E definitely fixed. There are some live stains, especially with fluorescence, as you might already know. But for the most part, we're talking about a fixed specimen, generally speaking. Okay, and the word stain, what is that? It's a general word. Again, biologists, bear with us, right? It's usually, we're talking about a chemical like amatoxylin, eosin, methylene blue, which you used on your cheek cells. Um, Technically, fluorophores are kind of a way to stain cells, so you could sort of push it and say that they are stains, but no one refers to them like that. Usually when people talk about stains, they're talking about something that isn't fluorescent, but it's loose, okay? It's a loose category. It means we're coloring our stuff, we're painting. We're adding contrast basically, but contrast through color of some sort. And there's a lot of chemicals that have been used for stains for a variety of reasons through the course of history. But let us now focus on IHC. So IHC is actually three things for our purposes. And as you'll see, the terminology gets a little loose and messy. But what I really want you to learn and understand are this slide, basically. The rest of this is explaining this slide. So there's IF, which is immunofluorescence. Then there's IIF, which is indirect immunofluorescence. So same thing, except indirect. And then there's IHC itself. And this reminds me a lot of bright field versus, that slide where I tell you bright, well, you might not have gotten it this year, but bright field includes the technique, the imaging modality of bright field includes DIC, phase, dark field, and also bright field. Because IHC is both the general category and also a type that is, like there's three kinds of IHC. One of them is IHC. I didn't make this up. It just has evolved that way, if you will. Okay, so let's look at the first type. So immunofluorescence. This is a key word, immune system. There's lots of things that are part of your immune system: cells, T cells, B cells, other cells, natural killer cells with all sorts of cool names, but also antibodies. And so in this case, the immuno part refers to the fact that we're going to use antibodies. And the antibody is going to have a fluorophore attached to it. So this is a chemically conjugated fluorophore that is attached to an antibody. And actually, as I say this, I'm sorry, I hope I'm not confusing you, because I just realized the example I gave you, however, is not an antibody. So, for instance, phyllocyanidin binds to actin. So phyllocyanidin is a molecule found in a mushroom that kills you if you eat it. Well, it kills you because it binds to actin. Actin is the protein that is in all of your cells, but especially muscle cells. It's just tons of actin. That's what you're eating when you're eating meat, basically. So for example, you can have phyllocyanidin, which exists in nature. It's a poison, but we get little doses of it in lab, not enough to kill anybody. And we conjugate it chemically to PHTC, which is a fluorophore. And so then you just add that to your specimen and it's going to bind to the actin specifically and not to anything else. So you're going to see where all the actin is and you won't see anything else. And all the actin will be a nice bright green. it will fluoresce green. That's an example of immunofluorescence, even though technically, again, I didn't make up these names and fields. Technically, we didn't use an antibody there, but you could alternately, and I use the example of phytsphalloidin almost unconsciously because it's the one that is done constantly all the time. Every lab that does IF and IIF, aka IHC, will have just a big, large stock of phalloidin in various colors. If you're multiplexing, right, you're doing multiple colors on your slide, then you might need a red phalloidin or a green phalloidin or rarely a blue phalloidin. So it's cheap, it's easy to get. It's why not? Let's see what the actin's up to, because the actin just sort of shows you the shape of each cell, since it's just a cool molecule. And since it's everywhere in the cell, it's a good way to kind of see like the whole extent of the cell. All right, so if it were a different molecule, let's say tubulin, which I have later in this lecture, you would have the antibody, Fitzy conjugated to the antibody to tubulin. So the antibody or some other molecule binds directly to the thing that you're trying to image. So the protein that you're trying to look at, actin's a protein. So I want to see actin. So I'm going to get something fluorescent that also binds to actin. So there's two parts to what you're deploying against actin, if you want, what you're staining actin with. Well, it's one molecule, but one part of it is a fluorophore, and the other part of it is an antibody, or maybe this famous molecule called phalloidin that directly binds to actin. It just loves actin. But let's say there's nothing like that for tubulin. So if you want to look at tubulin, you're going to have have an antibody that maybe has a red fluorescence tag, and then you can co-stain these two. You can see actin and tubulin both, which is actually a very common thing to do. So hopefully that is not too confusing, but basically immunofluorescence is you're tagging your molecule of interest by using something that specifically binds to that molecule of interest. And that something that you're using is fluorescent. So it has two functions. One, it fluoresces so you can see it. And two, it binds to whatever it is you're trying to see. So it's kind of like, putting a fluorescent spotlight on your protein of choice. Now, what happens a lot is instead of doing IF, we do IIF, which sounds like a song, actually. IIF. Anyway, indirect immunofluorescence. So it's the same thing, except it's indirect. Why indirect? Well, because this time one antibody wasn't enough. We're going to use two. There's a primary antibody first, and there's a secondary antibody, the second. AB, by the way, means antibody. And it's the second one that's fluorescent. The first one only has a simple job to do, which is bind to your protein of choice, your molecule of interest. So let's say you're trying to bind to tubulin. That's a protein that is found in cells. It's part of what's called the cytoskeleton. So this little skeleton that holds up cells. It's also all over, also very pretty. You're going to see it later, I think. So there's a protein in your cells, and you want to see that protein and no other proteins. How are you going to see it? That protein is not colored, it's not fluorescent on its own. You're going to see it because there's antibodies that a mouse has made. Somebody injected tubulin into the bloodstream of a mouse, and then the mouse made antibodies because they were like, what is this molecule in my blood? Why is it in my blood? Especially if it's, let's say, human tubulin. The mouse is like, eh. And the immune system, when it sees something weird, it makes antibodies to it. Yes, same as we're talking these days with COVID. There's different kinds of antibodies. Antibodies themselves are proteins, and you know, some of them actually mess with the thing that they're like, if there's an antibody to some of the spike proteins, you know, on COVID, you might have heard some of the the proteins that COVID uses to attach itself into cells. And so antibodies can sometimes actually like interfere with what's going on. And sometimes they are just basically post-it notes for your immune system, that your immune system, some cells of the immune system basically slap on this new invader, new protein that they haven't seen in the body yet. And it's telling the rest of the immune system to destroy that. So that's the real life world function, you know, the physiological function of antibodies. Here we've isolated them. We're just exploiting them or using them in lab. And so we inject, for instance, a human tubulin into mouse blood. And then we literally just take some of its blood, because a couple of weeks later, it has made lots of antibodies to the tubulin. It's like, I don't recognize you, be gone, protein that I don't know. So because antibodies circulate in the blood, it's really easy to make them, easy to get them, easy to know if they exist, etc. So that's how we make, for instance, primary antibodies, is we just inject them, in fact, into mouse or rabbit or something like that. And if we're lucky, it doesn't always work, but if we're lucky, the mouse, the bunny, or the rat will start making antibodies to our protein of interest. And then we'll have what's called a primary antibody to that. So we will tag the cells, tag our specimens with that primary. But that primary, you might notice, is not fluorescent. We still can't see it. So what if there's an antibody? But here's the cool thing. We have an antibody to that antibody. That's the secondary antibody. The secondary antibody recognizes the part of let's say, mouse antibodies that is that all mouse antibodies have. All antibodies have this Y shape, and so they have parts that are always the same and parts that are different. The parts that are different are the parts that recognize the specific thing you're trying to tag with that antibody. But the parts that are the same are recognized by the secondary antibody. So you have ready to go in your lab a bunch of secondary antibodies, probably made in a goat. This goat was injected with some mouse antibodies, and the goat's immune system made antibodies to all my mouse antibodies, which is fine. The goat is happy it doesn't have autoimmune disease or anything, right? Because it's recognizing there's weirdly mouse antibodies in my body. Okay, immune system, destroy them. Before that happens, we've gotten some blood, and then the goats go live their best life on a farm. They are not eaten or anything. So it's actually kind of not a terrible thing to do to an animal. They're not sick from it or anything, in case you're interested. Okay, so that's how we make secondary antibodies. And then we take them to the chem lab and we add the fluorophore to the secondary antibody. So now we have something fluorescent that can bind to the primary antibody, which bound to the protein of interest. And as an example, the slides that we have from molecular probes, I think you have gotten a chance to use them in this class or see them, but you'll see an example later. All right, so that's IF and that's IIF. IHC. What's IHC? Well, it's this whole category of things, but it's also, people will use it to just directly refer to something when that secondary antibody is not fluorescent, but there's a color changing molecule attached to it. So it's conjugated to color changing molecule. And that molecule is often something that turns brown. There's enzymes involved and it turns brown. Or I think there could be different enzymes. It could turn blue, for instance. And you may have experienced this by doing an ELISA, either in a lab class, because they're kind of popular to do, or you know, at home. For instance, pregnancy tests are ELISAs. So you may have come across an IHC without, you know, an example, at home IHC. You may have even performed it without realizing you were doing it. ELISAs are really common, really helpful all over the place. All right, so let's talk about this terminology. What is what? There's IHC, IIF, and IF. I just gave you the best definition I could figure out after all these years of asking people, teaching, and looking it up. It, however, in truth, they're used kind of interchangeably. And, so if your new boss uses it wrong on the first date, don't correct them. Basically, just learn what the culture of that lab is and how they refer to it. Fortunately, it's often in separate industries. And so until I started teaching histotex, I didn't realize that they had their own version of IHC. They were talking to me and I was confused about what they were saying, and I had to finally figure it out because I was like, IHC is fluorescence in my world. And they were talking about DAB and brown and showing me pictures that had nothing to do with fluorescence, and they'd never been on a fluorescence scope. And anyway, that's how I figured it out, that it's used very differently in different worlds. There's also ICC, just in case somebody brings that up. That's immunocytochemistry, which is basically the same thing as IHC. It's only depends on who. I haven't figured out who uses that yet. You can let me know. You can report back from your jobs when and where that is used instead of IHC. Obviously, histo means tissue and C means cell, cyto is cells, but we're loose with our definitions. And yeah, IIF and IF definitely use fluorescent scopes. I haven't heard of it in any other use other than fluorescence. But those of us who use fluorescence also call it IHC. When I'm doing IIF or IF, it's a mouthful. And for some reason, IHC is easier to say. I don't know why, it just is. And so we call it IHC. But if a histotext says IHC, they're probably talking about the enzymes and color changes that are visible in transmitted light scopes. And I have to say, histotexts kind of are right when you think about it, because they are starting with tissues, not just a layer of cells or an embryo. So if anybody should own IHC, it kind of should be them or you guys. But us research biologists and biotech folks, like I said, we use it broadly also, even though I guess technically we're doing IIF and IF. And I know that now there's in diagnostics and histo labs, there are people doing IF, right, or IIF. Usually IF because it's quicker, but it just depends. All right, so let's look at pictures, because picture is worth all of those words, right? Here's a classic example of IHC as done by in the histo world version of IHC. So you're going to get the brown is tumor. Often you're looking for tumors, or wait, in this case, this might not have been this looks pretty non-tumor-like to me as I'm looking at this tissue. I think they were just using the brown to look at the expression of a protein called P53. It is it's involved in a lot of tumors. When it's misregulated, it's a protein that has to do with the cell cycle. And so, if it's overexpressed, if there's too much of it, it can be indicative of tumors. But I'm looking at this beautiful regular tissue going, I don't know, that looks pretty normal to me. But anyway, the point is, when you see brown like this, that's usually IHC. So somebody has already done the regular H&E stain, and then now there's an antibody involved, because then they added an antibody to this tissue, to this specimen, this slice. And then there was a series of steps, and basically you ended up with that antibody showing up where there's the brown stain. There's enzymes involved. And DAB is this brown chromogen, as it's called. It's basically something that makes it colorful. Okay, so there's a typical IHC slide. Now that you've probably seen a lot, certainly in a histo class, you're going to see them. And here's an IHC slide as per a research biologist or a biotech person would think of it. But really, it's actually IIF and IF. It's both in this slide. Side note, notice there's some nuclei that, I don't know if you can see the arrow there, saturation. Ah, and this is in the promo materials. They're trying to get you to buy this slide, which used to be $50, now is $213 to my shock. But these are prepared slides that Molecular Probes, which is the company that is the source of all the good fluorophores, basically, sells for people who just need a beautifully triple stained slide to use, for instance, in teaching. um, and so anyway, it's just nice. And Molecular Probes was bought out by Invitrogen, which was bought out by LifeTech, which is bought out by Thermo Fisher. Anyway, so you can verify the price if you want to look at it here. So in this, green is tubulin. So as I promised, you're going to see some tubulin. That's the green. Tubulin actually is all made at one place near the nucleus. So you sort of see this characteristic spot where it starts and then it branches out. I'm looking at this and I don't know how it's showing up on your screen, but I gotta say on my screen, this is not the best image I've ever seen. You guys, I can take better images than this, but anyway, it'll serve the point of here's a triple stained slide with red for actin and blue as usual is the nucleus. Okay, so there's IHC, which is really IIF and IF both. I'll show you later which one's which color's which. And then just going back for a second, this is IHC as in true proper IHC, I guess, still with antibodies, but also with enzymes. And this is with antibodies, but with fluorophores instead of enzymes for the color. All right. And note that all of this was on fixed specimens, okay? None of it was live. So that's an important point when you're thinking about science and certainly about diagnosis. So what are the components? Let's go over that a little bit again. So an antigen, AG, is any molecule that has an antibody that binds to it. Is very circular definition because an antibody is something that binds to an antigen. An antigen is something that an antibody binds to. Basically, not all molecules out there in existence are antigenic. So we can't always get an immune system of whatever animal we're talking about to make an antibody for it, to produce an antibody. It's pretty amazing. The whole story of antibodies is extraordinary because we have just a few genes and they make just a huge, insane variety of thousands of different antibodies to thousands of different molecules. It's kind of crazy. And when you were a baby and when you were an embryo inside your mother, your thymus gland screened all these possible all these possible, antibodies that you might ever make. And threw out any that would attack your own tissues and left any in reserve that we don't know what they are, so they must not be us. And then later on, if you got sick, you deployed some of them. And then since this is something that might happen again, you made a whole reserve army of, these antibodies in case you saw this strange thing again, and you could tell your immune system to destroy it. I hope you realize I just described vaccines, right? That's how vaccines work. They get you to they get you prepared with a whole bunch of antibodies ready for whatever is you're vaccinated against. Okay, so I strayed from the topic a little bit, but it's hard not to talk about, vaccines when we're talking about antibodies and antigens. Okay, so antigen, as it turns out, for instance, COVID, the second COVID, SARS-CoV-2 is highly antigenic. The other one's not so much. So we got lucky and unlucky with that, basically. Unlucky because, of course, it killed so many people across the world. Lucky that we were able to make a vaccine to it. All right. So antigen is anything. Anything could be an antigen, any molecule. Usually it's a protein, though, almost always. It's actually part of a protein. You can have multiple antibodies to a certain protein. They're just binding to different parts of that protein. An antibody is a protein for sure, because it's something that is made by your cells, by your immune system cells, by B cells specifically. And the magic of an antibody is that it is super specific and it will bind to only that antigen. In fact, only that corner, if you will, of that particular antigen. Um, is it 100% completely specific? No. When you're doing IHC, you actually have to make sure it's not binding everywhere else too. I mean, there's a lot to be said for the right antibody and the right dose, and there's better antibodies and worse antibodies and so on. Um, Anytime I go to a conference, it's amazing to me how many companies out there are selling antibodies, and not all of them are good. So these are antibodies for research, right? Then, so you really have to, when you're doing IF or IIF, your primary antibody is so important. Do you have a good one? Does it bind strongly? Is it really specific? Sometimes you want to study something, a certain protein that you've discovered, and you're like, oh, okay, let me see where it is in the cells. And it's hard to get that primary antibody. Developing a primary antibody is a lot of work. Um, and you know, if you have a good one, then you can go to town and just do a ton of research, or even, you know, diagnostics. You can just learn so much from it. If you don't, then um, you know, plan C, D, E, F, something, you know, whatever. You have to figure something else out. Now, the secondary antibody is an antibody to an antibody, right? They're from different species. So the primary and the secondary have to be from different species, if you think about it. Otherwise, the whole scheme will not work. And the secondary antibody is lab created, like I said. Goats and donkeys and creatures like that are donating a little bit of their blood and the work of their immune systems to us for that. So let's see, let's look at some pictures, well, some schematics. Here's IF. You basically have an antigen and then you have an antibody. So the antibody is this Y-shaped thing, a little green Y-shaped thing. The arms of the Y are the part that literally recognize and bind to the antigen. The little stem of it, if you will, is the part that is the same in every single antibody of a species. So all of our antibodies as humans have the same, it's called the FC, anyway, the same, like there's a part of the antibody that is the same for all humans on the whole planet, for all of our antibodies. And then there's a part of the antibody that's super specific for that particular antigen. And then in this case, because we're doing lab work and we're doing IF, we're adding a fluorophore. So we're using chemistry to add a fluorophore to an antibody. This is, of course, not something that happens in your actual body on its own. So antibodies exist, but your antibodies are not fluorescent. Neither are mine. But we can create a lab situation where we make antibodies fluorescent. And we have that lovely primary. So basically, if the primary antibody is fluorescent, you just stick it on. It's IF, done and dusted, easy peasy, just one step. As you saw from my example earlier, sometimes in IF, the thing that you're adding, that you're using, is not actually an antibody. It's something like phalloidin, which happens to stand in for antibodies because it binds so well to actin. So we just made the phalloidin fluorescent, and then anytime we want to see actin, we use that. Okay, so, but generally IF is an antibody with a fluorophore, and that antibody is specific for whatever it is you're trying to see, which is now called an antigen because you've got an antibody involved. So sometimes what you're trying to see is better seen with IIF. I'll tell you why in a second, but here's what IIF is. Basically, you add a second step to it. You add another antibody. So there's the antigen, and then you still have your primary, but this time the primary is not labeled with a fluorophore. So it's sticking to your antigen, but you can't see it yet. So you call out your secondary antibody to the rescue. The secondary antibody sticks to the primary antibody, not to the specific site, but to the species part, the little bottom part of the Y that is the same for all species. So for instance, if your primary was made in mice, your secondary is going to recognize all mouse antibodies, but it has to be made in something else, like a goat. So it gets all kinds of fun because you've got your goat secondary, which is, you know, made in goats, but against mice, or made in donkeys against rats, made in really goats and donkeys mostly, made in goats against, did I already say rats? Against bunnies, whatever. So there's all these species involved in you getting an image, basically. So you have in IF, you have your antigen, you have your primary, which binds to the antigen, you have your secondary antibody, which binds to your primary antibody, and it's your secondary that has the color on it, the fluorescence, the fluorophore. So why would we do IIF? Why not just do IF? Like, why are we going to even bother with the secondary? Why are we adding complication and time and things that could go wrong? Why a whole different step? Well, for one thing, the secondary antibody is super cheap. It's in the 30s, even now, I think, like for a nice fat vial of it, instead of several $100s for a good primary antibody. So you can buy it in all colors and have it ready to go. And you can buy it in all species and sort of all combos and have it ready to go. Another reason is that by doing the second step, you have an amplification of the signal, because although I've drawn it as though there's only a secondary antibody binding to the primary, in truth, there's going to be a whole bunch of them. And so everything just got brighter. And as we know, brightness is important. Most of what we're looking at is super dim and we're trying to see it. And so having more signal gives you more resolution, more everything, more info accuracy, etc. And then it's just convenient because once you sort of wrap your brain around all these convolutions of, you know, go to anti-mouse, etc., and it becomes second nature to you, you realize that it's so easy if you just have a bunch of secondary around, and then you can design so many experiments with them, and you're sort of ready to go really quickly anytime you want to do something new. So it may be counterintuitive when you first encounter this and you're like, this is unnecessarily complicated. You know, why are scientists always more complicated than they have to be? But actually, once you start doing it, you kind of realize this is genius design. It's just very flexible, and you can make all sorts of interesting combos, because for instance, if you have a primary and if you're doing IF, you're stuck with the color that you have. You're usually not going to get through primaries in 5 different colors because it's so hard and expensive to get just the one primary. So like this guy is always going to be green, but what if I wanted to multiplex it to co-stain it with another green, another primary head and green? I can't ever. But if I'm doing IF, I can make that antigen any color I want it to be, because I just have a variety of secondaries that I can deploy. Okay, so, you know, and just making sure I set everything on here. Yeah, so for instance, if you have a goat anti-mouse green, whatever green fluorophore, you can use it on any mouse primary. So you just buy your $30, you know, vial of goat anti-mouse Alexa 488, which is a green fluorophore. And then any mouse primary that comes along for the next 10 years, you can use, you can see, basically. So here's an example from before, same bad image of a multiply stained, you know, triplex. I just made that up. We say multiplex, but we never say triplex. So ignore my unhelpful making up of words. But anyway, triple stained is what we actually say, a triple stained specimen. So these are BPAE cells. Since you're doing tissue culture right now, they're bovine pulmonary arterial endothelial cells. So the thin cells that line the arteries in a cow's blood vessels in their lungs. Why? Why BPA cells? Because they're pretty and they grow well in cell culture. I don't know how somebody first figured that out. You know, somebody had some cow lungs, I guess, and the arteries, and ever since they've been growing happily, and they're very adapted to lab in the to life in the lab, and the nice TC incubator. Okay, so in this specimen, both IF and IIF were done. IIF is the green. So the tubulin, there's a primary that is mouse antitubulin. If you click on the website that I gave you previously, or you look at the previous slide with this image, I included the label. So you should look at that wording below. I basically decoded it for you on this slide, but that's exactly, you get these long labels like where they say, for instance, BPA cells stained with mouse anti-alpha tubulin Bodipi FL goat anti-mouse IgG. That's all a mouthful to say that the green is was IIF. Specifically what? Well, there was a mouse antitubulin. So tubulin is what we're looking at. And the primary was made in mouse. And then their secondary was made in a goat against mouse antibodies. Bodipi is the fluorophore that happens to be green and has a very cool acronym. Okay, so that's it, 2 steps. First step, antibody against tubulin. That primary happened to be made in mouse. So our secondary is going to be an antibody against mice antibodies that happens to be made in goats and has a fluorescent tag, which happens to be a green. There we go. That's how we got the green. And we can see the tubulin, which is part of microtubules, which is what makes up something known as microtubules. So small tubes that are inside cells. IF, the red. The red is straight up phalloidin. You've heard of phalloidin before because I was talking about it. It binds to actin. So I should have really written that. But this is how my brain went. Phalloidin, we know that's actin. So actin, ACTIN, it is a tiny, tiny, oh, they don't even say it in the, look at that. They don't say in the label either because everybody knows phalloidin, that means actin. So actin is, you know, again, meat, protein, it's something that's in all cells. Okay, so we're looking at that with the red, and Texas red happens to be the fluorophore. So in this case, it's just one thing. It's phalloidin that has Texas red attached to it. The phalloidin is going to bind to actin. The Texas red is going to allow you to see the actin. And then blue almost always is DAPI, which just binds to DNA and it's fluorescent. So it's just a one-step thing. As I was making this slide, I noticed it said nuke blue, and I'm like, I have never heard of that before. But good job trying to sell me something new, which is exactly why they didn't use DAPI, because everybody uses DAPI, and they're just trying to, you know, invent a new product, which if I had more time, I'd check it out. Okay, so notice that you can do IIF and IF in the same protocol on the same cells. And in fact, you often do. You almost always are doing a direct stain for DNA, which most of the time you're just doing it so you can see the nucleus. If you're going to do anything, if you're going to stain for whatever it is, while you're in there doing the protocol, you might as well just throw in some DAPI at the appropriate time and see the nuclei, unless for some reason you're trying to see 3 things that aren't DAPI. So basically, if you have a channel, you have a lane. If you have an open channel for, you're trying to multiplex and see up to 3 colors. Some people even do up to 5. And you only have 2 things you're looking at, then just throw in some DAPI. And, the nuclei are nice to kind of orient yourself in a cell, because each cell should have 1 nucleus. Usually they also tell you a lot about cell health, because, there's a look to nuclei. Are they a certain even size and shape, or are they starting to look weird? All right. So but here's a big part that I haven't really mentioned yet. I would say it's, one of the struggles. Antibodies are big old proteins. They're not gigantic, but they are proteins. They can't just waltz in across the cell membrane of a cell in a live cell. The whole point of a cell membrane is to keep things out. It's fat, it's oil. Um, Protein's not just going to waltz across it. You proteins need to be chosen and escorted in live cells. And the live cells are not going to bring in a fluorescently tagged antibody. They're going to be like, are you freaking kidding me? No way. So the only way you can get an antibody inside a cell where it needs to be to bind to actin or tubulin or even the DNA is kill the cell. So an embalm it, basically fix it, put some fixative on it. So freeze time, stop it in that moment in time. And then you're still not done. It's fixed. Fixed just means that everything is chemically cross-linked. And so things are frozen in place where they are, we hope, as they were in life. Nothing has moved. It's literally like sort of saying, freeze to the cell. Okay, don't move, nothing moving. Um, doesn't mean that but you can't get the antibody in because the plasma membrane is also fixed, it's intact. So now you've got to poke holes in the plasma membrane that are big enough for your antibody to come in. So here's a graphic of that. And what poking holes is called in actual science lingo, it's called permeabilizing. So you're making the membrane permeable. That means things can go in and out, as opposed to impermeable, which means it's doing its job and not letting anything in. Um, there's all these mechanisms to check if the cell wants to bring something in, and how it's going to bring it in and so on. So an intact membrane won't let antibodies just waltz in. How do you permeabilize a cell membrane? With soap. Okay, soap is really good at destroying a membrane, at destroying lipid structures, basically. So you fix it with fixatives like formaldehyde and embalming fluid, basically, small version for cells, just a lot less of it. And then, so you fix it and then you permeabilize it. These are really important steps because you have to think carefully about them for your particular tissue, your cell, for what you're going to do. Fixatives are often fluorescent, which is an issue. So you might have to use methanol instead, which doesn't fix things as well. There's a lot of knowledge and art and skill towards having a good protocol and executing it correctly. So I'm summarizing some really important steps that have to happen for immunohistochemistry, IAF and IF. And this is true in all of them, basically. You have to fix the cells and then permeabilize them. And then now these little Ys are your antibodies. Those are just primaries because you'll notice that they don't have fluorophores attached to them. So probably I'm going to next have to throw in another antibody with a fluorophore attached to it. Okay. So I guess actually I've drawn IIF, the first step. So again, you fix the cells, you poke holes. Those circles are holes in the plasma membrane is the idea. In that cell, that's just one cell we're looking at from the side. And then, now that there's holes in the membrane and everything's frozen and stopped, you add the antibodies and they go in and they're going to bind to the particular molecule of interest, their antigen, which might be tubulin, or might be DNA, it might be actin in the three examples I've given you, but there are hundreds of thousands of proteins, so it could be a lot of other things. And then you're going to you're actually going to do some washes in between all this, but in a few other steps. You're going to actually block sites that love to soak up antibodies first. So there's, again, more details to this, but this is the general idea. And then, of course, if you're doing IF, you're going to add a secondary antibody with a fluorophore, so it fluoresces. Or if you're doing the classic IHC, you're going to add a substrate for an enzyme that will basically turn brown. Or if it's an ELISA, same thing, only a substrate, an enzyme and a substrate that happens to turn blue and sometimes other colors. So for this class, I want you to understand well everything I've said up to this point. There's a lot out there on the internet, videos, share it with each other, you know, Wikipedia, of course. There's a lot of info about all this. What I try to give you is the gist of it in a overview so that when you encounter it, you're ready to go. You have sort of a big picture idea of it, and then you can dive into the details in a lab when you're doing this. I'm not going to hold you responsible for knowing the following protocols, but I want you to see them because, and it's just really good because a protocol tells you what you're doing, right? It's everything. And these aren't specific protocols with amounts and things like that, but they're just the general steps that you do. And I've sort of summarized these already, but here it is. You're going to fix the cells, for instance, with formaldehyde, which cross-links proteins. You're going to permeabilize them using soap to poke holes in the plasma membrane. You still have some lipids left. The cell's sort of opaque still, but it's kind of a shredded plasma membrane, okay? It's fixed, but it's got holes. And then you're going to add a blocking agent, which is going to sort of just go around and lightly cover things that might just really grab onto any old antibody. And there's a lot of washing steps in all of this. You're just always constantly, whatever you put on, you're going to put it on for a limited amount of time, and then you're going to wash them out. The times are usually 10, 15 minutes to an hour. Sometimes some things take a little more time than that. But then you're going to finally add your fluorescent primary antibody, or maybe your fluorescent phalloidin instead of an antibody. But you're going to add your tag basically, or your stain. You're going to add something fluorescent that specifically binds to whatever it is you're trying to see, to your antigen. And then there's going to be washing. There's always more washing with PBS. And finally, at long last, hours later or days later, just depends, you get to visualize and see what you're looking at on the fluorescent scope. IIF is the same exact thing, except you'll notice, so you fix cells, you permeabilize, block, primary antibody, wash again. I say wash, but actually you're doing like 2 to 3 washes, just to make sure that you basically, while you're washing, is to wash away things that aren't stuck or things that are lightly stuck. You only want those primaries that are really hanging on they've really found their match, their perfect antigen, and they're just sticking like glue because they love that antigen or whatever, they recognize it. Okay, now you're going to add your fluorescent secondary antibody. Again, you're going to wash and make sure that, you know, you're only leaving behind things that are really stuck to that primary. And then you're going to, again, visualize it. So one extra round of basically letting it sit in the dark and maybe an hour or something like that, plus some washes. So you're adding an hour and a half to your protocol. IHC, well, really the same general thing. You're going to fix. And then depending on what you're doing, you may have to permeabilize or not. But then you're adding the primary, the secondary, and now you're adding the secondary has, for instance, DAB on it, but then you have to add something to make that color change. So when you're using enzymes, there's actually another step. It's not just fluorescent. There's actually yet another step where you're adding something to molecularly change the substrate that you've got attached to the secondary antibody. But they're quick, it's instant. It's an instant reaction. So it feels quicker to do IHC. And then you're going to look at it on a transmitted light scope and look for the brown precipitate, or you're going to, if it's an ELISA, well, I think I have that. So let's go see. Oh, and this is an interesting link. It's just, if you look at AbChem IHC, it just came up. I thought it was a good explanation. This is the bigger picture for histotech. if you're doing IHC, first there was somebody who grossed the biopsy tissue, made it into smaller chunks, got rid of the fat, all that. And then the tissue had to be fixed, you know, in a tissue processor. You embed it in paraffin, you cut it into thin sections using a microtome. What I don't have on here, I really should. You probably actually did an H&E stain. And then you did the IHC on top of it. So then you had primary, secondary visualizing reagents. And then you saw it on a transmitted light scope. And actually the pathologist probably is the one who looked at it to make the diagnosis. So just the workflow in a histolab. And and this is just H&E alone, workflow, same start to it, and then stain it and visualize it. So basically you take this H&E, you do that, and then going back a slide, then you do the IHC thing on top of it, in addition to the H&E. I don't know that I've ever seen IHC without H&E, because you want to see the rest of the tissue. So you're always going to do an H&E. You're always going to stain it that way. Since this is kind of a little bit general in terms of specimen prep, focused in on HC, but of course, just a note that histotechs sometimes freeze things in OCT, cut them into thin sections using a cryostat, and then fix and then stain them and visualize them. I super love cryostats, because I think it's amazing that, you know, there's a patient open in surgery and the surgeon sending you tissue to see if they can close them up, if they've found the margins of the tumors. And I think everything histotechs do is amazing, but this is one of those, like, I just fangirl around this. I'm like, and you have the magic power to, in 15 or 20 minutes, make a beautiful slide that, you know, tells somebody, you know, tells the surgical team, yes, you have taken out the whole tumor. So it's so cool. All right. What about an ELISA? I mentioned to you that technically an ELISA is a type of IHC. But again, nobody ever talks about it that way. They just talk about it, and ELISA, it's its own category, because probably it's so big in terms of usage. Like, it's so widely used by a million people. It's just such a great technique. It's so versatile for many uses, but it's really the same exact thing we just talked about. You're going to fix the cells, you're going to be a permalyze them, blocking agent, primary antibody, secondary antibody, add a color change enzyme. This time it's not into brown, it's usually into blue, or these days pink or whatever. And then you're gonna, well, if it's an ELISA that's meant to be used by a lab, you're gonna look at it on a transmitted light scope. Actually, often on slide scanners, you're often doing this in 96 well plates, where you're looking at large amounts of data. And there's robots basically are doing the ELISA, and literally robots, like robotic machines. And then they're also, then you throw it into robot microscope, which is a slide scanner, which is going to look at all those 96 wells and go, yes, no, yes, no, yes, no, reacted, didn't react. And you're trying to scan through large amounts of data. You're asking some kind of question where you're looking at thousands and thousands and thousands of things. Like, you're trying to see if something binds to something, maybe because then it could be used medically, or whatever question you're using. ELISA is usually a high throughput technique, where it's not like a technician doing one ELISA at a time. It's robots doing thousands and thousands and thousands of ELISAs. I mean, not that it can't happen that you do a personal ELISA in a lab, or a few ELISAs, but it's just so widely used in industry a lot too. And then it's also used for pregnancy tests, as I've mentioned. So there are ELISAs where you don't have to fix and permeabilize your urine, obviously. the ELISA is recognizing something that is already available in your urine, which is a hormone that happens to be there. So there's not a cell wall, a cell barrier. You get to skip those two steps, because you're like, I don't remember fixing any cells. I don't even remember any soap. And that is true. You're skipping ahead to the part where you've just got an antibody and a color change reaction, basically. And so it's used HIV tests, but also COVID quick tests or ELISAs. ELISAs, by the way, are quick and easy, but also totally unreliable, which is why, those quick tests there's a lot of false positive and false negative rates with an ELISA. So for instance, with HIV, for instance, when they figured out the ELISAs for that, it was a big breakthrough, right? It helped testing a lot. But then, so then we had like the quick and easy tests that are done with, just go in, spit on something, uh, and and you get results for that and a lot of other things. Any test like that, that's like 15 minutes to a result, or in the case of pregnancy tests, 2 minutes, is probably an ELISA. And then often what you do is you run a Western blot, like for something like HIV, for instance. That is a better test. It's more specific. It's less prone to errors to confirm if it was, whatever it is. And so the same for COVID quick tests versus, running a better test. COVID tests right now are mostly PCR, which is more accurate than ELISA, but takes longer. It takes about an hour or so, but then it takes a while to get it to the lab and back and so on. There's a lot of people developing saliva COVID tests and things like that, all sorts of, and even just breathing out on a sample, on a, test kit tests, and it's kind of cool. Anyway, ELISA is used a lot for all sorts of things. And you can read more about it at Wikipedia. And then just to remind ourselves, sometimes we get to look at live specimens, and we do that mostly by using GFP and fluorescent proteins. And it's an entirely different workflow there, because you've got to do something called transfecting, which means adding a plasmid into your target cells. So getting your target cells to accept this little piece of DNA that plasmids were invented by bacteria to trade genes and basically be able to resist antibiotics and things like that. We use them to, carry genes around. And for instance, a gene with your protein plus a nice little fluorescent protein tag at the end of it. And an interesting side note, though, that's kind of significant, fluorescent proteins, of course, are wonderful because we can get movies, we can get live cells doing live things, but they're really hard to fix. So you sort of have to choose, do you want fixed or live? And yeah, you're doing mild, sort of a low level of genetic engineering. It's actually quite easy to do. Like often you just literally put the plasmid plus something called lipofectamine on the cells and voila, they go in. It's highly specific. Fluorescent proteins, of course, led to this imaging renaissance about 20 years ago that led to the Merritt microscopy program existing and the kind of jobs that we train you for. And this is just my little drawing. That's the same cell, only now I've drawn in the nucleus. And there's step one, transfect with a plasmid. That can be a 15-minute thing in a few days to, let it incorporate, or it can be a lifetime of struggle depending on your plasmid and your cells and so on. So that's either very easy or incredibly difficult. And then here's an example, just like you've got there's a video of these are like little feet, basically, little attachments to the substrate to what the cells are on. Those are 3 cells. And they're a set of proteins known as focal adhesions in live cells. So that it's just from an old class where I did an example. So in this case, the protein of interest is something called vinculin instead of actin, tubulin, or the nucleic acids like DNA that like I've been talking about. There are things that are fluorescent dyes beyond the fluorescent proteins, but they're a little kind of dying out. in use because they're a little hard to use, but some are still around. And of course, as you know, some specimens are autofluorescent, so they just present themselves to the scope and they're like, image me. You don't have to do anything. I make fluorophores, like the original fluorescent protein, which is, you know, jellyfish make them and corals make them, scorpions, plants, lots of things are just autofluorescent. And these days cameras are sensitive enough so that we're actually able to use autofluorescence to look at vitamins. Vitamins in our cells are autofluorescent. So are like those vitamins that we eat every day. So sometimes your cheek cells fluoresce because they're coated in that. That happened when you're in the program. It took us a while. Amy Choi was a student, like figured out what was going on because some people had autofluorescent cheek cells. And I was like, they can't be. Like, that's not possible, but they are. Um, and so those people were eating, they were taking their like mega dose vitamins right before they scraped their cheek cells. All right, so some things are autofluorescent, and actually diagnostically, this is starting to be used because, for instance, it's called the extracellular matrix, collagen and things like that, that are made by cells, but outside of cells to give tissue structure. The ECM in tumors is quite autofluorescent. And so there's a project where this has been working, but not approved in the US yet. They have a tiny little confocal, well, the arm, like just a little part of it, scan a person. They were doing arms for a long time. So they'd scan your arm, just shine light on it. And it was able to see actually deep into the dermal layer. So where I was like, oh, 300, microns, they sort of engineered it so they can see a little bit deeper than that. And they can look at um, skin tumors before you can see them visibly on the surface, so they can catch skin tumors early. That's just an example of using autofluorescence. And it's sort of a contrast to all this work that we do with primary antibody and secondary antibody with a fluorophore washing and blocking and all this other stuff to do IIF to get something to fluoresce. Sometimes things already fluoresce on their own. And in mammalian cells, so humans and other animals, there wasn't much that was fluorescing, but as our techniques got better, we're now able to actually see metabolic products and vitamins and ECM of tumors, and some things are starting to get to that level. Well, they were always there. We are getting to the level where we can see those things, and maybe we can do it live and not do IHC. But nonetheless, IHC, including IIF and IVF, sorry, IF, are going to be around for a long time because they're super useful for so many uses. And here's an interesting point to think about. If you see a fluorescent image, you have no way of knowing whether it was live or IIF. So was it a fluorescent protein? Maybe it was a fluorescent dye. Maybe it's autofluorescent. Maybe it was fixed and stained and it was IIF, or maybe it was fixed and stained, permeabilized, and IF, aka IHC. But you're looking at a fluorescent image of cells. Just because it fluoresces does not tell you how the specimen was prepared. Unless it's a movie. If it's a movie, it's not IIF, right? Because that's fixed. So it can never be IIF, IF, IHC if the cell is moving, if there's a movie. What people do, of course, is do like grow cells out and then fix them at different time points. So we do that a lot. We're like, let's add the treatment, the hormone, whatever it is we're doing to these cells. Here's time 0, here's half an hour later, here's an hour later, here's 5 days later. So you sort of take time points in different sets of cells. So if you're watching the same set of cells or tissue or embryo or whatever it is, whatever the specimen is, and you're watching it change over time, it's a movie of it, then and it's fluorescing, then it's a fluorescent protein, a fluorescent dye, or autofluorescence. And I think that is it. These are some links that I used in making this lecture, or some interesting links for you to check out. So I hope you have enjoyed this glimpse into all the many ways that we can make specimens, including IHC, whatever that means to whoever you're talking to.